Bismillahirrahmanirrahim. Today's lesson we will study or we are talking about the detection of carbohydrates. Okay, detection of carbohydrates. What detection of carbohydrate means? It means to assess carbohydrates or to study some characteristics of the carbohydrates and to examine some samples to detect is there a or is this samples containing carbohydrates or not. Okay, so this detection will go through some tests or simple tests we're going to do or we're going to study now. The first one is solubility test of carbohydrates. Solubility test of carbohydrates. So this test principle depends on the solubility of carbohydrates in water. Okay, this test make us to know the various of solubility of different carbohydrates in water, that according to their or depending on their chemical structure. As you know, carbohydrates are organic matter or organic molecules containing carbon, hydrogen and oxygen. So it contains many many hydroxyl groups. These hydroxyl groups enable it to dissolve in water, but depending on the chemical structure of the carbohydrate, as we will see. Some carbohydrates are soluble or has high solubility in water, and some of them are less solubility in water, and another group of carbohydrates has no soluble in water. Okay, so the solubility test of carbohydrates is an important procedure used to assess how different carbohydrates dissolve in water. Carbohydrates, depending on their chemical structure, show varied degrees of solubility. That means there are different degrees of solubility depending on their chemical structure, as I said. This test helps in understanding their role in biological system, food science and metabolic process. Okay, the objective of the test is to test the solubility of different types of carbohydrates in water and classify them based on their solubility. So we are going to do to test these different types of carbohydrate solubility. So we will imagine that we have 3 samples or 4 samples to test. Each one are carbohydrate, but each one a different type of carbohydrate. We will test each sample to know the degrees of solubility of each one, and then we will classify them based on their solubility. The materials: carbohydrate samples. The samples we will use is to test glucose, sucrose, starch and maltose. Okay, as you know, glucose is a monosaccharide, sucrose is disaccharide, starch are is polysaccharide and maltose is disaccharide. Okay, distilled water, we need distilled water, test tubes, stirring rod or vortex mixer, heat source, which is optional for polysaccharides like starch, need some heating. Okay, so proceed the procedure. Number 1, prepare samples. Take a small amount, 0.5 or 1 gram of each carbohydrate. Each of carbohydrate samples, you must take 0.5 to 1 gram and place it in a separate test tube. That means we will have 4 test tubes for each sample. Okay, 1 for each one. Okay, and we will place 0.5 to 1 gram from each sample and place it inside the test tube as in the picture. And we must label each tube, okay, to know the sample or to identify the sample. The step number 2, add water. Pour around 2 to 5 ml of distilled water into each test tube. Okay, we're adding 2 to 5 ml in each test tube. Number 3, stir the mixture. Stir the mixture. Mix the contents using a stirring rod. Okay, using a stirring rod, and I think you know the stirring rod or vortex mixer. I will show you the picture of vortex mixer. It's a device, simple device for mixing. Okay, so we will going to mix the contents using a stirring rod or vortex mixture. Then observe the behavior of each carbohydrate in water while you are mixing, or after you mix them all. Observe the behavior of each sample in water. Heat if necessary. That means some of these samples need heating, but not all of them. So which one need heating? The starch. Some carbohydrates like starch may require heat to dissolve or form a suspension. If testing starch, if you are going to test the starch, heat the mixture in a water bath for better results. Okay, so that means starch will have no soluble or does not soluble in water, in cold water, but it needs heating to dissolve. Okay, the observation. Observe after stirring or heating. Observe the solubility. Soluble, we're going to say this sample is soluble if the carbohydrate dissolved completely in water, forming a clear solution. Okay. Partially soluble if the carbohydrate forms a colloidal dispersion, like starch in cold water forms a suspension. Insoluble if the carbohydrate does not dissolve and remains as a solid or undissolved particles. Okay, so this test I had attached you to in the platform. I had attached the video from YouTube to see the practical of this test to see the solubility of each sample. The result interpretation. Number 1, monosaccharides, example glucose and fructose, they are highly soluble in water due to their small molecule size. As you know, glucose is monosaccharide, so it has a small molecule size and polar hydroxyl groups. Okay, so glucose and fructose are highly soluble in water. Disaccharides like sucrose and maltose, they are soluble in water, but some may need stirring or slightly elevated temperatures. But are soluble, but some may need stirring or slightly elevated temperatures for better dissolution. Okay. Number 3, polysaccharides such as starch and cellulose. These are generally insoluble in water, but starch, however, may form a colloidal suspension or gel upon heating and can dissolve in warm water, especially when treated with iodine. Okay, so when testing starch, when heated with water, starch form a paste or gel. Iodine can be used to test for the presence of starch, as it forms a blue-black color with iodine. Okay, the summary of these results. Summary of solubility of common carbohydrates, which are tested. Glucose, highly soluble in water. Sucrose and maltose, soluble in water. But starch, insoluble in cold water, but forms a gel or suspension when heated. Okay, you can go to platform and check the linked for a video in YouTube and watch the test, the solubility test. Okay. Another test to detecting carbohydrate is Molish test. Okay, this test depends on to detecting the presence of carbohydrates in some samples, to know if these samples contain carbohydrates or not. Okay. So Molish test is a chemical test which is used to check for the presence of carbohydrate in a given analyte. This test is named after Australian botanist Hans Molish, who is credited with its discovery. So it is discovered by Molish. So the name comes from the person who discovered it. Molish test involves the addition of Molish reagent, which consists of solution of alpha, naphthol, and in ethanol, to the analyte, and the subsequent addition of a few drops of concentrated sulfuric acid to the mixture. Okay, we are going to illustrate more about this test. So the Molish reagent we will add. It consists of solution of alpha naphthol in ethanol. This is Molish test. This is Molish reagent. We will use this reagent on Molish test. And also we adding a few drops of concentrated sulfuric acid to the mixture. The formation of a purple after adding Molish reagent and drops of concentrated sulfuric acid. Then after formation of a purple or purplish red ring at the point of contact between the sulfuric and the analyte and Molish reagent mixture, confirms the presence of carbohydrates in the analyte. So the positive test, or the positive result for the Molish test will be after presence of, or after formation of a purple ring or a purplish red ring appear in the junction or the point of contact between the three layers of sulfuric and analyte, which is the test sample, and the Molish reagent. After and the point of contact of these three substances, that confirms the presence of carbohydrate in the analyte or in the sample. Okay, so we will see the ring, purple ring. That mean this is positive for Molish test. That means there's a carbohydrate in this sample. If there is no ring appear, or there is no purple ring appeared, that means the sample doesn't contain carbohydrate. Okay, the principle of the Molish test. In Molish test, the carbohydrate, if present, undergoes dehydration upon the introduction of concentrated hydrochloric or sulfuric acid. As you know, sulfuric acid makes dehydration for carbohydrate. That means take the water from substances. That resulting in the formation of an aldehyde. Okay, this is the equation, or this is the illustration of what happens. Okay, we can say again, the carbohydrate, if present, that means if the sample contains carbohydrate, undergoes dehydration upon the introduction of concentrated hydrochloric or sulfuric acid. That means when we add sulfuric acid or hydrochloric acid, concentrated hydrochloric or sulfuric acid, that resulting in the formation of an aldehyde, because the dehydration of sulfuric acid and or hydrochloric acid. This aldehyde undergoes condensation along with alpha naphthol present in the reagent, resulting in the formation of a purple or reddish purple colored complex. In step 2, in the reaction, aldehyde undergoes or condense with alpha naphthol. What's the alpha naphthol? It's the reagent of Molish. We said Molish test consists of alpha naphthol in ethanol, right? So the alpha naphthol, when react to aldehyde, undergoes condensation, resulting in the formation of a purple ring or reddish purple colored complex. Okay, the procedure of the test. Number 1, in dry and clean test tube, drop 2 to 3 of Molish reagent. 2 to 3 drops of Molish reagent must be added to a small amount of the analyte or in the sample in the test tube and mixed well. So the first step is to put the analyte or the test sample in the test tube and add 2 to 3 drops of Molish reagent and mix it well. Number 2, a few drops of concentrated sulfuric acid. must be added dropwise along the walls of the test tube. We put drops of concentrated sulfuric acid on the walls of the test tube to facilitate the formation of a layer. Okay. Now a few drops of concentrated sulfuric acid must be added dropwise along the walls. We add it at the wall of the test tube to facilitate the formation of layer and avoid mixing. Okay, there is no mixing here. Try to avoid mixing, that to facilitate the formation of the layer. When you add sulfuric acid, don't mix, just to observe, or just wait to observe the formation of the purple ring. Number 3, the development of a purple ring at the layer formed by the concentrated acid is a positive indicator for Molish test. So that means when the purple ring appear or formed, when this purple ring formed by the concentrated acid, so that means it's a positive result for Molish test. And also that means there is a carbohydrate inside the sample. Okay, if no purple or reddish purple color arises, the given analyte does not contain any carbohydrate. Okay, that means this is a negative result for Molish test. If there is no purple ring formed, that means the analyte or the sample does not contain carbohydrate. Okay, or that we can say it's a negative result for Molish test. Okay, this is the picture you can observe in your left hand. This is a positive result for Molish test. That means this sample containing carbohydrate, because there is a purple ring formed in the test tube. And in your right hand you can see one of these two test tubes has no purple ring appear. That means it's a negative Molish test. So this guy here in this sample, carbohydrates absent, or there is no carbohydrate in this sample. And also you can see the positive Molish test. So a blue or purple ring appear in the test tube, so carbohydrates present. And this test also I attached to you on platform. You can go and check and watch the video of Molish test to see the practical. The result: all carbohydrates, monosaccharides, disaccharides and polysaccharides except tyroses and tetroses should give a positive reaction for Molish, and nucleic acids and glycoproteins also give a positive reaction, as all these compounds are eventually hydrolyzed to monosaccharides by strong mineral acids. Pentoses are then dehydrated to 4-phoral, while hexoses are dehydrated to 5-hydroxymethyl 4-phoral. So this 4-phoral and 5-hydroxymethyl 4-phoral, which react to the reagent of Molish, which is alpha-naphthol, and confirm the presence of sulfuric acid, concentrated sulfuric acid, and form the purple ring. Either of these aldehydes, if present, will condense with two molecules of alpha naphthol to form a purple colored product. Okay. Okay, the third test to detecting carbohydrate is Anthron test, called Anthron test. Okay, what's the Anthron test? Anthron test is a group test for carbohydrates that provide a rapid and convenient method for quantification of carbohydrates that are either free or bound to any lipids or proteins. That means you can detect carbohydrates as a free carbohydrates or bound to lipids or proteins, such as glycoproteins or glycolipids or glycoproteins. You can detect it all. Objectives of Anthron test: to detect the presence of carbohydrates in given solution. So you can detect if it's present or not, and to quantify the concentration of free and bound carbohydrates in a solution. That means to know the concentration after we check the presence of carbohydrates in the test tubes or in the sample, so we can detect the concentration of these free or bound carbohydrates in the solution or in a given solution. The principle of Anthron test: if carbohydrate is present in the form of free carbohydrate as poly or monosaccharides, or also bound as in glycoproteins or glycolipids, the concentrated acid in the Anthron reagent first hydrolyzes it into component monosaccharides. Similarly, the concentrated acid then catalyzes the dehydration of the monosaccharides to form furfural from pentosis or hydroxyl furfural from hexosis. The furfural or hydroxy furfural formed condenses with two molecules of naphthol from the Anthron reagent to form a blue-green complex. Okay, so the reaction will be between the furfural and hydroxyl furfural, which result from the hydrolysis of polycarbohydrate or monosaccharides. They had been hydrolysis to furfural or hydroxyfurfural. Then this furfural or hydroxyfurfural form condense with two molecules of naphthol from the Anthron reagent. So the Anthron reagent consists of naphthol to form a blue-green complex. So the positive result will be after form the blue-green complex. The complex can then be quantified by measuring the absorbance of 620 nanometers wavelengths in a spectrophotometer. So we will use the instrument spectrophotometer or in a red filter colorimeter. So I also attached to you a video to know how to use the device, a spectrophotometer, and how it works. Okay, so these spectrophotometers can quantify or can to detect the concentration of these carbohydrates in each sample. Okay, by what? By measuring the absorbance of light of or 620 nanometer wavelengths in spectrophotometer or in a red filter colorimeter. Okay, this is the equation, this is the reaction. We have here the glucose, okay, and then we add the sulfuric acid. Sulfuric acid takes water, so this result in hydroxymethylfurfural. Hydroxymethylfurfural comes from glucose because it's hexoses. That means glucose have 6 carbon atoms. Okay, so the result after adding H2SO4, which is sulfuric acid, the result is hydroxymethylfurfural, and then this react with the anthron reagent and make a condensed bluish green complex, and this is the positive result for anthron test, which appears the color bluish green complex. So that means there is a carbohydrate in these samples. So after we confirm there is a carbohydrate in the sample, then the next step is to know the concentration of each carbohydrate in the sample by using the device spectrophotometer. Reaction, hydrolysis of polysaccharides to monosaccharides is step 1. Hydrolysis of polysaccharides to monosaccharides. So if the sample is starch or cellulose, these are polysaccharides, right? So when we add sulfuric acid, concentration of or concentrated sulfuric acid, the hydrolysis of polysaccharides to monosaccharides will be the first step, and then monosaccharide will dehydration to furfural. And then okay, well, the step 1 is to hydrolysis of polysaccharides to monosaccharides, and then dehydration of monosaccharide to furfural or hydroxymethylfurfural if it comes from the hexoses like glucose, for example. So furfural or hydroxymethylfurfural reaction with an napsol in the reagent, in the anthron reagent, and then form the blue-green complex. This is for the positive result of anthron test. So if the result is negative, so there is no formation for blue-green complex. So that means there is no carbohydrate in the sample. Requirements, materials required. We have equipment, UV spectrophotometer, vortex mixture, mantle, heater or water bath. Okay. The chemicals or reagents we need: anthron reagent, glucose, other carbohydrates if desired. If you want to test other carbohydrates, you can do, and sample. Glassware and other equipments: test tube stand or rack, pipette, beaker, ice, ice to cold the samples, as I will show you. Test tube caps is to cover the test tube. Tissue paper and wash bottle. Okay, these are pictures of a UV spectrophotometer. As you see, there is a screen, or we can connect it with a computer. Okay, spectrophotometer make a graph to the result of absorption of light. Okay, so you will read the concentration of the carbohydrate samples through this screen in the spectrophotometer. And if you watch the video of you in the YouTube, which I link, which I attached to you, the platform, you will know how it works. And this, in your right hand, there is a device called vortex mixture. Is to mix the samples. As you see, you put the test tube, the sample in test tube, and put it over this, or put it on the, or install it in the vortex mixture to mix it. Okay, reagents. Anthron reagent. 2 grams of anthron is dissolved in a liter. This step to prepare the reagent of anthron. Take 2 grams of anthron is dissolved in 1 liter of concentrated sulfuric acid. Okay, the freshly prepared reagent should be used for the assay. So when you want to test, you have to use the fresh one, okay, by adding 2 grams of naphthol, so it's of anthron, dissolved in 1 liter of concentrated sulfuric acid. Okay, then we have glucose stock solution, 200 microgram glucose per ml distilled water. Dissolve this 200 microgram of glucose in ml of distilled water. Stock solution of glucose is to be prepared from the stocked solution. So this is stock glucose. We will take the samples from this stock. Okay. Note, other carbohydrates of same concentration can be used as samples if desired. You can use another carbohydrates, but of the same concentration of this stock solution. Okay. Procedure of anthron test. Number 1, pipette out different volumes, 50 microliter, 100, like this, 50, 100, I like this, and so on, of glucose solution. You take different volumes of glucose solution from the supplied stock solution which we prepared into a series of test tubes. Okay, and make up the volume to 1 ml with distilled water. Okay, take a tube labeled as blank. So this blank tube containing just 1 ml of distilled water. So there is no glucose here in this tube. There is no carbohydrate in this tube. It's just distilled water. So we will mark it as blank, or label this tube as blank. 1 ml of just distilled water, and the rest of the tube labeled 2 to 9 for the concentrations of a standard curve. The other tubes you make, and you know the concentration of them as the step of number 1. We take different volumes, 50 microliter, 1 microliter, and so on. So this tube, you know the concentrations of them. You will label them by 2 to 9. Okay, for concentration of a standard curve. This fall using in a spectrophotometer. Tubes 10 to 15 are for the unknown samples. Okay, there's unknown samples. Mark them with from 10 to 15. Add 5 ml of the anthron reagent to each tube. Each tube of these, we will add to them 5 ml of anthron reagent and mix well by vortexing. Then cool the tubes. And step number 5, cover the tubes with caps on top and incubate at 90 centigrade for 17 minutes, or boiling water bath for 10 minutes. Or we can go put them on water bath or in boiling for 10 minutes. Okay, and then cool the tubes to room temperature and measure the optical density of the solutions at 620 nanometer against a blank. So this after you have a positive test, positive result for the test in some tubes labeled from 2 to 9, which are, the concentration of glucose in each one. Okay, on these tubes you will measure the optical density to know the concentration of carbohydrates inside these samples. Okay, measure the optical density of the solutions at 620 nanometer against a blank. Prepare a standard curve of absorbance against glucose concentration, which is by the spectrophotometer. Determine the amount of glucose in the unknown sample by plotting a standard curve of A60 620 on the y-axis and concentration of glucose on the x-axis, and also this for spectrophotometer. Well, the procedure of anthron test, we can stop at the point of if there is a presence of carbohydrate or no. Okay, or it's a positive by forming the green or the bluish green complex or not. And then after the appearing of the color, or after we confirm the positive result of anthron test, then we can go to examine the concentration of the samples in the spectrophotometer. So the steps of 6 to 8, it's for the examine the concentration of the samples of the carbohydrate samples in spectrophotometer. Okay, result and interpretation of Armstrong test. The presence of a blue-green complex indicates the presence of carbohydrates in the given solution. Okay, if there's a presence of a bluish-green complex, that means it's positive result for Armstrong test. That means the carbohydrate is present in this sample. From the graph, which prepared by the spectrophotometer, we can determine the concentration of unknown samples. There's some unknown samples. You don't know the concentration of carbohydrate in them. So you can examine the concentration of each one of these unknown samples by put them in the spectrophotometer. In this picture, you can see the procedure. The sample of them here is cellulosic nanofiber, which is this white substance in the dish, and also they add sulfuric acid, okay, in the reagent, and you can see the solution confirm, or its solution formed is bluish green complex, which is a positive result for Armstrong. So that means there is a carbohydrate in these samples. Uses of Armstrong test. Armstrong test is used for the detection of quantification of carbohydrates in various samples, like blood, serum, milk, and its variation. Okay. Limitations of Armstrong test. Armstrong test gives a negative result in samples that are carbohydrates like D-glucose, phenyllozazone. It's hard word. Phenyllozazone, D-glucose phenyllozazone, and D-glucose phenylozotriol. Okay, these two carbohydrates give a negative result for Armstrong test because they do not form furfural or hydroxyl furfural in the dehydration step. Okay. I think it's finished for today. I hope you go to the platform and watch the videos which I attached to you to get more information after you see the practical test of each one of these tests. Yeah. See you next in the see you next fine, and bye bye.